So today we'll be using SpongeBob to better understand a molecular technique in molecular biology known as Southern Blot. In order to understand this technique, we must understand what molecular biology is. Well, molecular biology is a science that looks at how small biological molecules affect life. Molecular biology is used to understand how life develops, and molecular biology is everywhere. It's inside jellyfish, pineapples, coral, animals, plants, humans, and one way that we study molecular biology is through a molecule known as DNA. DNA is one of the most stable molecules on the entire planet. DNA is double-stranded and it has 4 base pairs. The base pairs A and T always bind with each other, and the base pairs C and G always bind with each other. DNA is an extremely large biological molecule that contains instructions for the growth of life, and DNA is specific for everyone. So my DNA is different than your DNA, and DNA is everywhere. DNA is inside me, it's inside you, it's inside fish, and it's inside animals, and we can use DNA to study how life develops. A Southern blot is a molecular technique that uses molecular biology to recognize DNA. And since DNA is specific for everyone, we can use DNA to identify different individuals. Let's say that one day over the weekend, you notice that someone ate a Krabby Patty in your restaurant, and you want to know whether it's Plankton or Patrick. Luckily, you take out your microscope and find saliva on the Krabby Patty, and it's important to know that saliva has DNA. Since we know that DNA is specific for every individual, we can use a Southern blot analysis to determine whether it was Plankton or Patrick who ate the Krabby Patty. So we're going to isolate a sample of saliva and place it in a test tube. Then we're going to use a DNA kit to remove and extract the DNA from the saliva. Then we're going to use a Southern blot to analyze the DNA. The Southern blot technique will take the large piece of DNA and cut it up into smaller pieces, and each piece will be known as a DNA fragment. The DNA fragments will be separated by size. Then, the DNA fragments will be matched up to a complementary sequence of DNA from either Patrick or Plankton. If Patrick's DNA matches up with the DNA fragment, then we know that Patrick was the one who ate the Krabby Patty. But if Plankton's DNA is the one that matches up with the DNA fragment, then we know that Plankton ate the Krabby Patty. And this process can take quite a while.
2,000 years later.
Okay, so the process won't actually take 2,000 years, but it can take quite a while for your DNA fragment to match up with the DNA of interest from either Plankton or Patrick. Luckily, molecular biology was able to help us uncover who ate this Krabby Patty. So basically, the Southern blot technique can help us discover or uncover or identify a specific sequence of DNA. And since DNA is specific for everyone, this technique can be really useful